Till now, we have understood about the morphology and the history of cell biology. Remember, we call viruses as the particles, not the cell. Singer, S.J. Singer and Nicholson. Very important names to remember in biology. Now, the arrangement is fluid because of various macromolecules which are present. Endocytosis. Endocytosis is a process of bulk transport inside the cell, right? So the plasma membrane will actually pinch out like this and it will have the molecules. This is a challenge for my 9th graders to solve a PYQ of NEET examination based on what we have learned today. Hey guys, welcome once again to this interesting session of biology by Devika, and in this particular class we are going to talk about something very interesting, that is cell membrane. So what is a cell membrane? Why do we need it? Till now we have understood about the morphology and the history of cell biology, but in this particular session we'll understand what are the members, which membranes which covers the cell, right? What is plasma membrane, and then who told us what is the structure of plasma membrane and what it is like. So it is like fluid mosaic model. So what is fluid mosaic model? That is also that we are going to understand in this particular session. So here the components of cell, something we have covered previously. Components of cell are what? The cell envelope should be there. Cytoplasm, nucleus or a nucleide should be there. So nucleus in case of the eukaryotic cells, where we have a proper covering around, right? We have an organelle, we have a membrane, double membrane. Nucleus is a double membrane structure present in eukaryotic cell, whereas nucleoid in case of prokaryotic cells, where the DNA is just scattered into the cytoplasm. Any which ways, this should be there, right? Cytoplasm has to be there and cell envelope should be there. So among this should be components of a cell, if 3 are present, then only we call it as a cell. Remember, we call viruses as the particles, not the cell, right? So we are going to understand what is cell envelope and what it is made up of. So as I told you, eukaryotic cell and the prokaryotic cell, right? So all these cells have different kind of cell membranes as well, okay? They differ in their structure as well. So eukaryotic cells, U in biology means true, okay? U, every time you come to this word, U, remember that U means true. And karyote is nucleus, right? Similar way, in case of pro, remember that means primitive. What does it mean? It means primitive type of cell, where you have nucleoid, and here you can see nicely double membrane structure of nucleus which contains a DNA, right? So membranes which are covering the cells are different in different kind of cells. You know that. In plant cells, you also have an additional layer of cell wall, whereas we do not have cell wall. And thank God we do not have cell wall. Otherwise, if some aunt of yours or your grandma would have come and pulled your chicks, they would have crackled just like a leaf. But that doesn't happen, right? Why? Because we don't have cell wall. We are flexible, we can locomote and we don't need it. Whereas the plants, they surely do need it for its support, rigidity, mechanical support. Right? Now, first, we'll understand what is the structure of this cell membrane that we are talking. So let it be plant, let it be animal, let it be a bacterial cell, we need to see that whether there is a cell membrane present or not. And yes, definitely there is cell membrane present, although the composition of this might be different, but cell membrane is definitely there. Otherwise, we would have never called it as a cell, right? So this semi-permeable membrane which encloses all the cellular component, it is a semi-permeable membrane. Semi-permeable, half permeable, not completely permeable. So it actually regulates what can come in and what can come out, right? What can go out and what can come in. All those things are regulated. It is selectively permeable. Yes, you can use both the words semi or selectively permeable, right? That means it doesn't allow anything and everything to come in. Other thing is it is elastic, delicate and quite flexible. Yes, it's flexible. It's not rigid at all. Then it is made up of lipids and protein. What is the major composition over here? This is the diagram of your plasma membrane and major component of it is made up of lipids and protein. Now, where do you find it? Obviously, outside of the cell, because it is enclosing all the cellular membranes, all the cellular components inside it. Then, it encloses the cytoplasm and nucleus of the cell. Where in case of the plant cells, where would it be present? It would not be the outermost layer, but it would be present between the cytoplasm and the cell wall. So here you will have the cell wall, right? You will get a rigid structure over there, right? That would be the cell wall, okay? Then in between you will have the cytoplasm and a large vacuole, nucleus and the other organelles as usual, right? And in between over here somewhere you will find your cell membrane. So when I'm talking about cell membrane, is it the same thing? Plasma membrane? Yes, it is the same thing, plasma membrane. So in case of the plant cell, where do you find it? You find it in between of the cell wall and in between of the, where? Between of the cytoplasm. So cytoplasm, cell wall, and in between there you have the cell membrane. Why am I emphasizing on this? Because such kind of tricky questions do come in your science Olympiads and it is very important for your NEET foundations as well. Right? So yes, that's the structure over there. So you have it over here, right? So this is the stuff that we are talking about. Then, this is also known as plasma membrane, already told you. It protects the cellular components and it helps in transporting of substances. What does it do? It helps in transporting of substances. In further few lectures, I'll tell you how do the plasma membrane take parts in transportation. Right? In fact, the next lecture itself, you'll find that out. Now, there was these two guys, okay, Singer and Nicholson. Singer, S.J. Singer and Nicholson. Very important names to remember in biology because these two scientists have actually explained us what is the structure of plasma membrane in the year of 1972. Well, there were many theories given, many models were proposed. Okay, it could be a sandwich model, it could be some other model, but Singer and Nicholson were the first one to tell us it is a fluid mosaic model. So we are talking about the structure, right? And the first time they were observing it in the RBCs itself. Okay, so they are made up of a phospholipid bilayer. Bilayer meaning two layers. Okay, there are two layers and protein molecules are embedded inside them. Now, if you want to understand, just for example, this composition of lipid and protein will vary from cell to cell. For example, in our RBC, in human RBC, we have nearly 52% of protein and 40% of lipid, right? But it may vary from one cell to another cell. This is important for our NSO-ISO examination and hence mentioned over here, right? 52% of protein and 40% of lipid. Why RBC is important? Because this were the first cells where the plasma membrane structure was studied. Okay. Then they proposed, okay, this is a fluid mosaic model, isn't it? They said it's a fluid mosaic model. Now there are two words over here if you pay attention, fluid and mosaic. Hey guys, have you seen those tiles? Okay, the tiles usually nowadays we use it for the pavements or in the corridors and all. So there what you have the tile, right? And the tile have a different kind of patterns on it, right? Like stones which are used for designing, making design, right? and that is why we call it as a mosaic tile. Here also we are calling it as a mosaic. That means here something is making a mosaic pattern, and that is protein basically. Okay, so here you have a phospholipid bilayer. Phospholipid bilayer, how is it a bilayer? First of all, here you can see, I'll use another pen so that it is visible to you, right? So that is one layer over here, right? And then another layer. So on, clearly you can see. If I remove one molecule, okay, if I remove one molecule from here, what you will find? You will find that there is a head region, right? And this head region is hydrophilic in nature. Hydrophilic means what? Water loving. So it is a water loving, right? Water loving head region. This is a water loving head region. Hydro. Hydro means water. Philic. Philic means loving. Water loving. And then it has hydrophobic tails. You can see this tails, these are hydrophobic tails. They hate water. So that's why they are placed inside. So there is one layer from top and the other layer from down. And both the, if in case this you consider as the cell outside region, right? And you consider this as cell inside region, right? Both the sides have hydrophilic heads, and in between there is a hydrophobic talca layer, right? So now that is how the arrangement of phospholipid bilayer is done over here, right? Now we understood that these are the phospholipid bilayers. Here you can see them very easily as well now, okay? Now this phospholipid bilayer, you can see certain kind of proteins being inserted over there, right? So there are this purple color molecules that you see over here, right? These are proteins, okay? This pink color molecules over here, these are also proteins. What are we talking about? We are talking about structure of plasma membrane, right? So this base we have understood that it is made up of a lipid bilayer, and in that now the mosaic pattern is due to what? That mosaic pattern is due to this proteins, okay? So then what does it have? It have proteins, it have glycoproteins. Now what is glycoprotein? What is glyco and protein? Right? Again, break the word, you will understand. What is glycoprotein? Glyco, glucose, matlab ki carbohydrate, and protein. So when a carbohydrate is attached to a protein, it will form glycoprotein. So a carbohydrated protein would be called as glycoprotein. Why? Because addition of such carbohydrates to the protein give them a proper structure, make them active protein. Okay, that's the modification basically. Then we also have certain proteins which are on top. They are just placed on top, okay, and some of the proteins are going through and through. As you can see over here, they are going through and through from both the lipid bilayers, right? So there are two types of protein now: intrinsic protein and extrinsic protein. Extrinsic matlab they are just on the top. Intrinsic, they are going through and through, are par, right? Intrinsic proteins. It also has cholesterol, right? It also have cholesterol molecules embedded over there. So all of these things make a mosaic pattern over there, right? There are some integral different kind of alpha helical proteins also you can see. Alpha helical proteins are basically simple kind of proteins, right? Simple kind of proteins. And then you have the surface protein. I told you already, the extrinsic protein are also called as surface protein. Globular protein, hydrophilic heads of the phospholipid bilayer, and glycolipids as well. What are glycolipids, guys? Now you can judge by the name itself. Glyco, carbohydrate, lipid is lipid, fat molecules. So glycosylation also it is called as. So these are actually addition of carbohydrate to the lipids. So all this stuff you can find embedded in this phospholipid bilayer, which gives it a mosaic pattern. Hence, We call it as a mosaic model. Now, I'll explain you why fluid as well. We'll try to understand why fluid mosaic model. Now, the arrangement is fluid because of various macromolecules which are present. Okay, it is arranged in such a way that it has a fluidity, right? And such membrane, such property is called as plasma, right? So, owing this fluidity property, if a membrane has, we call it as a plasma. And that's why we call it as plasma membrane. Otherwise, we could have just simply called it as the cell membrane. Understood? Now, the fluidity is so well managed over here that the phospholipid bilayer molecules, right, they can actually shift from this place to another place, like this. Okay, they can actually flip also. They can rotate and shift their place. They can rotate in the same place, or they can just come from this position to another position, right? So it is not fixed, right? It is not fixed, and such thing is called as a mosaic fluid, right? Where the molecules are not going somewhere else, they are staying there, but they are changing their places. So they are capable of doing that. Not only that, you would be surprised to know there's a flip-flop also can happen. That means this layer can go up and the upper layer can come down, right? So like this. So there could be a flip-flop also if required of certain kind of, proteins which have to be exposed to the outer side of the cell. So that can also be done by the cells. Understood now why fluid? So now it is, basically justified why we agreed to Singer's and Nicholson's fluid mosaic model of plasma membrane, right? So because of this movement of lipids and proteins, we have fluidity of the membrane. That is why we have fluidity of the membrane. All right. Now this fluid nature, is it important? If it is, how does it help us? How does it help the cell? Right? So it helps in quick repairs, right? Because if molecules are rigid, okay, so it would be very difficult for another molecule to come and fit in. But because it is fluid, now gaps can be filled up easily, right? There won't be pores for longer time in the cell membrane, and we can heal. So quicker repair. Ability to fuse. One cell can definitely be fused with another cell. This can be done naturally in sometimes. Some cells naturally do that, or in labs, okay, in vitro conditions, we do this for hybridization techniques and all, right? So by sonegation methods, that can be done. Cell growth is possible because it is fluid, it can expand and the cell can grow. Cell division is also similar way easy, right? So now this will divide into two daughter cells. We have seen binary fission. Yes, contraction of the plasma membrane will happen and then cytokinesis will happen. Endocytosis. Endocytosis is a process of bulk transport inside the cell. Okay, we'll talk more about it in some other session. Then we have secretions. So we have this glandular cells like your salivary glands, right? So they do the secretion by exocytosis, right? So the plasma membrane will actually pinch out like this and it will have the molecules, and now these molecules, if they are produced inside and they go outer side and then the secretion is secreted out, that's called as exocytosis, and exactly opposite is the endocytosis. Formation of intracellular junctions can also be done. Okay, so what are intracellular junctions? These junctions actually connect one cell from another cell. Now in a tissue, every cell cannot function as an individual cell. In multicellular organism, you know that, right? Hence we need a communication which is formed by intracellular junctions with the help of plasma membrane. Now, my 9th graders, can you solve this question? Let's see. Select the correct statement from the following regarding cell membrane. Lipids are arranged in a bilayer with polar heads towards the inner part. Polar heads towards the inner part. Head, huh? Then, fluid mosaic model of cell membrane was proposed by Singer and Nicholson. Sodium and potassium ion across the cell membrane by passive transport. They move across by passive transport. Proteins make up to 60 to 70% of the cell membrane. Pause the video and answer it in the chat box. Let me see how many of you can actually answer this. Right? I hope you have given the correct answer, because we have just now studied that the fluid mosaic model was proposed by Singer and Nicholson. Okay, so now this is a challenge. This is a challenge for my 9th graders to solve a PYQ of NEET examination based on what we have learned today. Okay, so let's make our journey towards NEET from today. So the plasma membrane consists mainly of what? Proteins embedded in a carbohydrate bilayer, phospholipid embedded in a protein layer, proteins embedded in a phospholipid bilayer, या फिर protein embedded in polymer of glucose molecules. So this is 2011 guys, 2011 PYQ of CBSE PMT. PMT is pre-medical test, right? It is a PYQ of your NEET examination. If you have answered C, bravo, give a pat on your back, right? You have already scored 4 marks in NEET examination today itself. Imagine what can you do by attending these lectures. Thank you so much. Please hit a like button and do subscribe and share the video with your friends. See you, tata, take care, have a nice day.